we are having biological chemistry as a special subject for the TYBSc zoology. So biochemistry, so what we learn throughout the semester, it is somewhat basic concepts of biological chemistry. Then what are the chemical bonds, what are the different types of the chemical bonds, how they are useful for in the universe, and what are their significant. Then some concepts we are studying, carbohydrates, then basic concepts of the proteins, amino acids, lipids, enzymes, their kinetics and so on. Now we will start some basic concepts of biological chemistry and basic concepts of the bonds. So basically bonds, that is called as the chemical bonds. So what the chemical bonds are responsible for? So what is the bond actually? The bond means to link to each other. When two molecules or two atoms are trying to unite to each other and link together, that may be called as a bond. So basically bonds can be classified into two. That is a non-covalent bond and covalent bond. But rather than this, some are assuming that another is ionic bond is also somewhat separate than the non-covalent and covalent. And one more thing is van der Waals forces. Van der Waals forces. So there are basic four types of bonds, and since the bonds are responsible for linking two molecules or two atoms together, then we'll justify some what is actually meant by ionic bonds. So ionic bonds are may be called as electrostatic bond or electrovalent bond. So the bond between the ions or electrons results from the attraction between oppositely charged ions, and this linking of the ionized atom is the formation of ionic bonds. So in this case, any atom tries to maintain its stabilizing capacity either by losing its electrons or by accepting its electrons. Say for example, Na, sodium. So always we denoted as sodium Na plus, and Cl always considered as Cl minus. Why it is so? Because Na has one electron which is an arbitrary electron called as moving upon itself outer orbital, and whereas in a chlorine atom, Cl atom, 7 electrons are moving around the orbital, external orbital. So Na, since it has one electron which is moving at the outer orbital, it tries to maintain its stability by giving its electron, and at the same time chlorine atom tries to maintain its stability or stabilizing capacity by accepting the one electron from the sodium. So Na plus Cl minus, it will form NaCl. So this is the actually stable molecule. Just like that, covalent bonds are there. Actually, covalent bonds are the strong bonds. Basically, we will try to understand what exactly means by the covalency. So covalency is nothing but when the electrons are not transferred. In first case, ionic bonds, electrons are getting transferred from Na to Cl. So in this case, electrons are not transferred, but electrons are shared between two atoms. So when the electrons are not transferred from atom to atom, but shared between two atoms, so this hydrogen atom which has only one electron comes together with another hydrogen atom, and the two hydrogen atoms share the two electrons between themselves to form a duplet. So this is a stable structure and is called as H2 molecule. Say for example, this H has one electron, then another H has one electron, then by sharing the two electrons by themselves, it will form HH, two electrons are getting shared, and it will form H2, which is a stable structure. Similarly, in a Cl atom, chlorine atom, there are 7 outer orbital electrons can come together with another Cl atom, and by sharing one electron between themselves can form an octet, and it is called as a stable molecule of a chlorine atom. Chlorine molecule. Then there are some examples of peptide bonds. There are some examples of covalent bonds. Say for example, one is a peptide bond, then second is a glycosidic bond, and third one is a disulfide bond. So all the bonds are examples of the strong bonds, I mean covalent bonds. So we'll explain one by one how it forms. Say for example, we'll take example of the peptide bond. So what is a peptide? Peptides is nothing but the part of a protein, and we know that proteins are made by the union of a number of amino acids. So simply we are saying that amino acids are the basic unit of a protein molecule, and when amino acids are getting united to another amino acid and so on, it will form a big molecule of a protein. So we will have to study what exactly means by the structure of amino acids. The amino acid, since it is a basic molecule or basic unit of a protein, so it has to be stated that, so each amino acid, when we see the structure, so NH2, Cr, then COOH, and this is the R group, maybe. So R group, all the amino acids in the nature are found. We are naturally found. There are 20 amino acids, and all amino acids are having the same structure except their R group. So each amino acid having two groups. At the left side it has amino group, which is the basic group, and at the right side it has a carboxylic group, which is the acidic group. So this is the basic fundamental formula of amino acid. So this is the one kind of amino acid formula. Another, we get another amino acid formula, NH2, C, R, H, COOH. So when this is the first amino acid, this is second amino acid, when the two amino acids are getting united to each other, it will form a specific bond that we call as a peptide bond. So how it is formed? NH2, CR, H, CO, NH, then C, H, R, and COH. So which is formed by union of a two amino acid. In between them, CONH bond is formed. So when each amino acids are getting united to another amino acid, so there is a formation of peptide bond, and at each and every time there is a release of one water molecule. So CONH is a peptide bond. Then we again see another kind of a covalent bond, that is the glycosidic bond. So what is the glycosidic bond? So glycosidic bond is occurring between the two monosaccharides, that may be glucose, that may be fructose, that may be any monosaccharides we are studying. Then we'll take an example of how this bond is formed. Say for example, glucose molecules. Glucose is a monosaccharide. So when we see the structure of glucose molecules, this is one glucose molecule. Then we again take another glucose molecules for understanding how this glycosylated bonds is formed. Then so this is a H, OH, then H, OH, OH, H, then H, OH, H, CH2OH. So glucose has 6 carbons, it is a hectose. Then another glucose molecules, same thing, H, OH, OH, H, OH, H, CH2OH. Another, this is a glucose molecule. So when this two glucose molecules, two monosaccharides are trying to unite to each other, it will form a very specific linkage that is called as a 1,4 glycosidic linkage. Why it is called as a 1,4 linkage? Because when we count the number of carbon atoms in the glucose molecules, so this is the carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6. Similarly, same case, carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 6. So when these glucose molecules are uniting to each other, it will form a linkage between carbon number 1 of one glucose molecules toward the carbon number 4 of another glucose molecules. And when this linkage is taking place, there is always release of one water molecule at each time when they are getting an atom. So it will form this OH. Here H2O is released. So this is called as a 1,4 linkage. Not only linkage, it is called as a 1,4 glycosidic linkage. Since it is formed between the union of two glucose molecules, they are called as a glycosidic linkage. Then this is a kind of covalent bond or strong bond. Then another one is a basic example called as disulfide linkages. So what is a disulfide linkage? What is disulfide linkage? Because we know that so all the proteins are made by the union of a number of its basic molecules, that is amino acids, and we also know that there are 20 naturally found amino acids in the nature. But among that amino acids, one amino acid is very specific, that is called as cysteine amino acid, which has one sulfur atom on its body. And when the formation of proteins in the form of basic structure, that is a primary structure, then secondary structure, then tertiary, then quaternary. So what happens actually, these proteins are formed by primary to secondary, secondary to tertiary, or tertiary to quaternary. So in the primary structure, so there is only linking between the one amino acid to another amino acid. That means amino group and carboxylic group of two amino acids are getting united to form peptide bond. That is forming a simple basic structure of a protein, that is a primary structure. And what happens in the second structure? Additional, along with the peptide bond, additional bonding is taking place. Hydrogen bonds there are, and it forms alpha helix or beta carotene structure. Then additionally, in the third or tertiary structure, additional bonds are taking place in that, along with the hydrogen bonds, along with the peptide bonds. Additional molecules or bonds they are called disulfide linkages because as and protein structures are taking higher position, that is from primary to secondary, secondary to tertiary. So it becomes more complex and complex. So due to this structure, additional disulfide linkage is formed. How that disulfide linkage is getting formed? Because when that molecules are trying to fold and refold upon itself, cysteine molecules are getting near to each other, and since we are having the structure of a cysteine which has a sulfur molecule inside, when cysteine molecules are coming together very near to each other, the sulfur group of one cysteine and sulfur group of another cysteine, SH and SH group are both cysteines are coming together, and when S and S bridge is getting formed, so that is nothing but called as a protein disulfide linkages. Of course, along with this peptide bond, glycosidic bond, these bonds are also the strong bonds and which are helpful for maintaining their stable structure of the protein. Then we'll go some more toward the non-covalent bonds. So what is meant by non-covalent? So just we have seen that in a covalent bond, the electrons are shared between two atoms or two molecules, but here is no question of a sharing. Of course, there are non-covalent bonds and they are called the weak bonds. Good example of a weak bonds is the hydrogen bonds. Another examples are there are hydrophobic bonds are also. So how these non-covalent bonds are formed? Take example of water molecule. So when we take examples of water molecule, water, the basic molecule structure of water is the H2O. Say for example, this is a O, this is a H, this is a H. But in water, not a single molecule is present. There are a number of molecules of water are present in water, and they are trying to attack themselves by the very weak bond that is shown by the dotted lines. That is, this is H, and it will form again another water molecule. Just like that, when this another molecule of H is there, O, H, like this. So all these water molecules are binding together themselves with the help of very weak bonds, and that weak bonds is called as hydrogen bonds, which are shown in their dotted lines. So this is hydrogen bonds, and hydrogen bonds are the examples of the weak bonds. Then how that hydrogen bonds are taking their position in the water molecules. So polar molecules such as the water molecules have weak but partial negative charge at one region of a molecule. The oxygen atom in water, which has a partial negative charge, and the partial positive charge elsewhere, that is hydrogen atoms in the water. So thus when water molecules are close together, their positive and negative regions are attracted to the oppositely charged regions of nearby molecules. The force of attraction shown here as in the dotted lines is called as hydrogen bonds, and each water molecule is hydrogen bonded to 4 others. So this is the hydrogen bonds. The hydrogen bonds has only a 5% or so the strength of a covalent bond. However, when many hydrogen bonds can form between the two molecules or parts of the same molecule, the resulting union can be sufficiently strong as to be quite stable. So there are examples of multiple hydrogen bonds by which many molecules are formed and get stable. So multiple hydrogen bonds hold the two strands of a DNA molecule, DNA double helix together. They hold the polypeptides together in such a secondary structure as the alpha helix and the beta conformation. Then they also help enzymes bind to their substrate. They also help antibodies bind to their antigens. They also help transcription factors bind to each other, and they also help transcription factors bind to DNA. So another example of non-covalent bonds, just I told that hydrophobic bonds. So what is actually hydrophobic groups? Hydrophobic groups are the groups which are repelling to water molecules. So hydrophobic, water repelling group and water repelling group or atoms, are they getting united to each other? So that is simply called as a hydrophobic interaction. So they are called as non-polar interaction, or it may be called as unenergible interaction. So this interaction of a non-polar hydrocarbon molecules forced together because of a stronger water-water interaction. So there are some significance of hydrophobic bonds in the biology. So hydrophobic interactions are important for the folding of proteins. So this is important in keeping a protein stable and biologically active. So because it allows the protein to decrease in surface area and also reduce the undesirable interactions with water. So besides from the proteins, so there are many other biological substances that rely on hydrophobic interactions for its survival and functions. Say for example, the phospholipid bilayer membranes in every cell of your body. So along with this peptide bond, ionic bond, covalent bond, the non-covalent bond, so we are having some specific bonds. They are also weak bonds called as van der Waals forces. So van der Waals forces, we are having some functions of bonds. Why they are very significant to maintaining the life of biology or maintain the stable power of biological conditions. So I have enlisted some basic functions of the bonds here. Number one, the covalent bonds like peptide bonds, just we have seen earlier in the peptide bond, that is the CONH, are formed between amino acids linking total number of amino acids present in that protein and thus responsible for primary structure of protein biomolecule. Similarly, the disulfide linkage, that is the SS linkage, formed by covalent interaction of SH groups of two cysteine residues or cysteine amino acids. The oxidation of SH groups results in SS bridge formation that we called disulfide linkage. So these are strong bonds that give rigidity and better stability to the tertiary structure of a protein biomolecule. Number second, the non-covalent linkages like hydrogen bonds, hydrophobic bonds, and electrostatic bonds are weak bonds, but there are multiple interaction of this kind give the protein biomolecule characteristic fairly rigid three-dimensional structure. Then number three, the hydrogen bonds formed between purine pyrimidine bases, that is A double bond T and G triple bond C, are responsible for stability of double-stranded DNA helix. Function number 4, the covalent bonds like alpha and beta glycosidic linkages are responsible for structure of cellulose, starch. Starch is a very big polysaccharide which includes linear linkage of the glucose molecule, that is amylose, and branch linkage of amino acid, that is not amino acid, branch linkage of the glucose molecules, that is amylopectin, and by adding amylose and amylopectin it will form a big molecule, that is starch, which is polysaccharide, and one more is glycogen molecules, which is again polysaccharide. So number 5, hydrogen bonding plays a key role in determining the shapes of large biomolecules like proteins and nucleic acids. Number 6. Van der Waals forces are responsible for attraction between very close to biomolecules, result in the formation of, say for example, lipoprotein, that is lipid plus proteins, glycoprotein, that is glucose plus proteins, and nucleoprotein, that is nucleic acid plus protein complex in chromatin. Number 7. The hydrogen bonding is the important feature of the structure of the liquid water. Of course, we know that. Number 8. Ionic interactions occur between oppositely charged groups. Ionic forces may involve when a protein binds to another molecule. And number 9. Hydrophobic bonds are created indirectly by the effect of water in forcing hydrophobic groups together to prevent them from disrupting the network of hydrogen bonds in the water molecule itself. So thank you. Any question? No question? Thank you.